Hi, my name is Cameron,
And I'm Riley.
and today we're going to be talking to you about the enzyme amylase. So before we really dive deep into all of the information about the enzyme, we just kind of wanted to give a basic background, just to kind of lay the foundation for the presentation. So just to start off, amylase is found in the pancreas as well as the small intestine, salivary glands, and even the liver. However, the highest amount or the highest concentrations are found in the pancreas. And when amylase reaches the stomach, it actually becomes inactive due to how acidic the pH is. So further breakdown of the starch occurs by another form of the enzyme, by the pancreas, which is why the concentration is so high there. And just to kind of give a basic definition of what it does, it actually catalyzes the hydrolysis of starch into sugar products.
So looking a little more at the gene level, alpha amylase is 9,262 bases long. It is found on the plus sense strand, which means this is the strand that is transcribed into mRNA and then translated into proteins. It is located on chromosome 1, and here is a partial mRNA sequence that we found of the gene AMY1A.
So next we're going to go into a little bit of the breakdown of the enzyme classification. So the main class of amylase is a hydrolase, which is where the 3 in the enzyme commission number comes from, where it uses water to break a bond. And then the second class is a glycolase, which is where the glycosidic bond is hydrolyzed. And then the third is the glycosidase, which is where the enzymes catalyze the O-glycosyl compound. And then the last number of the EC nomenclature comes from whether the enzyme is in the alpha form, the beta form, or the gamma form. So if it's in the alpha form, it ends in 1. If it's in the beta form, it ends in 2. And then if it's in the gamma form, it ends in 3. So it's also made up of 496 amino acids as well.
So just a little bit of a general overview of the 3 forms of amylase. There is alpha amylase over there that is the major form in humans and other animals. There is beta amylase that is found in seeds and fruits. And there is gamma amylase that is found in trace amounts in the small intestine. Alpha amylase is found primarily in the salivary gland and in the pancreas. And alpha amylase is made up of 3 domains, A, B, and C. The A domain is the core domain of amylase. It is made up of a beta-alpha barrel, and it is also the domain that contains the active site. The active site is located closer to the C-terminal end, and it contains amino acids such as glutamic acid and aspartic acid. The B domain is made up of sheets of 4 antiparallel beta strands with long loops between them, and the C domain is made up of 8 beta strands that are assembled into a globular unit forming a Greek key motif.
And next we're going to talk mainly about the alpha amylase due to the fact that it is most pertinent to us as humans. It's actually the enzyme that's produced through the digestive system of mammals. And one of the key factors in amylase is calcium because it's essential for its function and it helps maintain the enzyme activity. And even in some cases, it can help the protein, or it can help the enzyme from becoming denatured due to heat, and that's one of the essential roles of it as well. And the optimum pH for alpha amylase is 6.7 to 7.0, so it is fairly neutral. And it can act anywhere on the substrate, and it's also known to be the fastest acting of the 3 enzymes compared to the beta form and the gamma form.
So the substrate for amylase is starch, and starch is a polymer of glucose that is linked by glycosidic bonds. There are 2 polymers, amylose and amylopectin. Amylose at the top is a linear polymer with up to 6,000 glucose units with an alpha 1-4 glycosidic bond. And amylopectin down here is a linear chain of 10 to 60 glucose units linked by an alpha 1-4 glycosidic bond, and then also a side chain with 15 to 45 glucose units linked by a 1 to 6 glycosidic bond. And amylase cleave the glycosidic bonds of amylose and amylopectin.
So now to go a little bit into the reaction that occurs here. So as Riley was saying, we have the amylose backbone at the top, and then we have the amylase along with water because it is a hydrolase, and this essentially rapidly, the enzyme amylase rapidly cleaves the 1,4 glycosidic bond linkage in the amylose portions of the starch. And because of this, this is where we get the breakdown of the two sugar products producing the maltose and the glucose.
So the primary source paper that we used was high endogenous salivary amylase activity is associated with improved glycemic homeostasis following starch ingestion in adults by Mandel and Breslin. So the objective of this paper was to determine whether increased digestion of starch by high salivary amylase concentrations can predict postprandial blood glucose following the starch ingestion. They predicted that individuals who produced more salivary amylase would have faster and more substantial postprandial blood glucose responses following starch ingestion than individuals who produce less salivary amylase. So it is assumed that high amylase individuals may be better adapted to ingest starches, whereas low amylase individuals may be at greater risk for insulin resistance and diabetes if chronically ingesting starch-rich diets.
So to start off, I'm going to go over the methods with you all. So the participants in this study were from Philadelphia, Pennsylvania. All of the participants had to have a BMI index of lower than 25 kilograms per meter squared. They also had to be healthy individuals, non-smokers, and they couldn't be on any type of medication that affected their saliva at all. So in a lot of cases, there's medications that have side effects that cause dry mouth, so participants on those types of medications wouldn't be able to participate because it could potentially skew the results. So there were also a total of 19 individuals who participated. There were two groups. There was the high amylase producers and then the low amylase producers. There was a total of 10 high amylase producers and 9 low amylase producers. And 14 of these participants were female, whereas there were only 5 males participating in the study. So as far as the actual experiment goes, there was two laboratory sessions that occurred in the morning. The experimental group was given 50 grams of a cornstarch solution, whereas the control group was given 50 grams of a glucose solution. So these participants, no matter what group they were in, they were not allowed to eat or drink anything besides water, starting at midnight the night before the study was conducted. And they also were, when they drank each solution, they had to drink each solution at a constant rate for 20 minutes, and the rate of intake was timed for experimental purposes. Additionally, they also collected saliva of the individuals. It was taken before drinking the solution, and it was put into tubes. The tubes were weighed both before and after the saliva was taken to determine the salivary flow rate, and then the tubes were centrifuged and then frozen for further analysis, which I'll talk about a little bit later. And then lastly, they did blood sampling as well in EDTA-coated tubes. So they collected blood 5 minutes before they drank the solution, and then at 0 minutes before, which is essentially right before they drank their specific solution. So the blood samples were obtained every 3 minutes for the first 15 minutes of the study, and then every 15 minutes for the next 2 hours. So in addition to this, they also assessed the participants' dietary intake, where they gave them a questionnaire so they could determine their starch intake as well as their fat and carb intake. And they also specifically were looking to see what kind of foods they ate that were high in starches as well. And then for DNA extraction, they used qPCR to look at the AMY1A gene, which is a diploid gene. and they specifically used the copy number determined by the TACAM copy number assay for the AMY1 gene. And then for data analysis, which Riley's about to go into the results, they used an ANOVA.
So looking at the results, this table shows the enzyme concentrations were analyzed by amount and activity to ensure that the difference between the groups was not simply due to differences in salivary flow rate. There was a positive relationship between the individual amylase concentration and the number of AMY1 gene copies, and so this shows that high amylase group had more AMY1 gene copies than the low amylase group. So this figure shows the postprandial plasma glucose concentration in adults by salivary amylase activity after starch ingestion. Following starch ingestion, the plasma glucose concentration differed over time between the two groups, and the high amylase group had low postprandial glycemic responses at 45, 60, and 75 minutes post-ingestion. So these figures show the postprandial plasma insulin concentration in normal weight adults by salivary amylase activity. So the plasma insulin concentration following starch ingestion did not significantly differ at any time point between the groups, but the high amylase group had increased insulin concentration at 9 minutes compared to their group baseline, but the insulin concentrations for the low amylase group did not increase at this time. So the results did show the opposite of their original hypothesis. The high amylase individuals had significantly lower postprandial blood glucose responses, mediated by the increased plasma insulin concentrations.
Okay, and then the last part of our presentation goes into the bioinformatics that we looked at. So we specifically looked at predicting antigenic peptides for the AMY1 gene. So we wanted to kind of relate our paper into the bioinformatics portion. So we looked at epitopes for polyclonal antibody production, and polyclonal antibodies are essentially a collection of many immunoglobulins that target different epitopes or binding sites for a single antigen. So when talking about the AMY1 gene, like we mentioned previously, there are 9,262 bases long for this, and there are 750 residues. But based on what we did with bioinformatics, it's determined that there are only 28 antigenic determinants, which you can see here that we included in the slide. So we wanted to thank you guys so much for watching, and we hope you enjoy our presentation on amylase.
Thank you.
And here are our references.